All right, you're good to go.
Okay, well, good morning or good afternoon, good evening, depending on where you're viewing from. My name is Mark Saltzman. I'm a professor of biomedical engineering at Yale, and I'm delighted to be able to talk to you this morning in New Haven, anyway. And I'm gonna talk about some of the work we've done over the past few years. My research group and I at Yale on biomedical engineering of delivery systems for gene repair. And I was going to confine it to just that subject, but I found when I was putting this together, I expanded. And so I'm going to include also some discussion about how we can use this technology for cancer treatment and for disease prevention. And I know you have access to a chat, so you can feel free to ask questions at any time. And I'll probably save the answers to the end. But if it's something that you really feel like you need the answer to quickly, let us know that through the chat feature. And my email address is here on the screen now, and also easy to find online. And so if you have questions afterwards and would like to contact me, please do feel free. I'm going to change the pointer here just so that it makes it a little bit easier for you to see. And I'm going to start by telling you a little bit about myself and how I got to Yale and the kind of things I do in addition to research and teaching at Yale, and then I'll launch into the talk. But I grew up in Iowa, in the middle of the United States. And if this was an in-person presentation, which I wish it was, then I would ask if there are any Iowans listening in, any Iowans in the audience, and I hope that there are. For those of you who don't know where Iowa is, there it is, right in the middle of the 48 continental United States. And this is a picture of my grandfather's farm in southern Iowa. And this is what Iowa looks like if you haven't been there. It looks a little different from New Haven, maybe different from where you grew up. It's a lovely place in many ways. So I studied, I went to college at Iowa State University and went to graduate school at MIT. So I moved to the West Coast. And then since I finished my PhD at MIT, I've been taught at a number of other universities, and finally ended up with my dream job at Yale University in 2002. So I've been here for 18 years. I've taught over the past several years a variety of courses. A course that started off in the first year seminar series, but then opened up to a wider audience called Frontiers of Biomedical Engineering, which was an introduction to the field. I taught a course on physiological and biological determinants of global health, sort of mixing global health needs, what causes states of unhealth, how that differs in different populations throughout the world, and what the biological basis of those differences are, and an introduction to the technologies that can be used to address global health problems. The main course I've taught, and I've taught it many times now, is a required course for biomedical engineering students, but it's also cross-listed in molecular, cellular, and developmental biology called Physiological Systems. It's a course on human physiology, and popular among students that have some interest in medical school, but really a fun course to teach, and one that I'm very proud of. And I've taught an upper level course on engineering of drug delivery systems, which is really the topic of my research group and the kinds of things that I'm going to be talking about during this presentation today. So I'll start with this question: why is Yale the best place in the world to be a biomedical engineer interested in medical devices? That's what I am. I'm a biomedical engineer interested in medical devices. It's the best place in the world for a variety of reasons: fabulous students and postdocs. So I hope you'll become one of the fabulous students that come here to study whatever it is you want to study. Hopefully some of you will want to study biomedical engineering, but you'll be surrounded by a terrific group of peers. And I've really enjoyed all my interactions with students and other scientists here. So this is a photograph of a recent photograph of some of the members of my laboratory group. I'm going to talk about one person in particular toward the end, Elias Quijano, who's featured here. Elias started working in my laboratory when he was at the end of his first year of Yale College, and continued there for many years. And I'll tell you a little bit about his story later. I have fabulous physician scientist collaborators, and I'm going to mention some of them as I go. Marie Egan, who's a pediatrician, Michael Girardi, who's a dermatologist, Peter Glazer, who's a radiation oncologist, for example, Joe Pietmeyer, who's a neurosurgeon. And it just creates a terrific environment for learning and for doing research in this field. And we have fabulous resources in engineering and medicine. The Department of Biomedical Engineering, which has grown over the years that I've been here, and is a remarkable place, and other sort of interdepartmental Center for Biomedical and Innovative Technology, where students and faculty can work on independent projects and sort of think about how to take them from the laboratory and make an impact in the world. So I'll start here the research part of the talk by just reminding you something you probably already know, that many of the products that make modern healthcare effective are innovations that arose from collaborations between physicians and engineers. And I'll just give you a couple examples of those. This is a dialysis filter for kidney dialysis. Blood comes in one end and goes out the other. The blood flows through this series of hollow fibers that are on the inside, and there's a solution that flows over the outside of the fibers called the dialysate. And waste products that accumulate in the blood when your kidneys aren't functioning properly are filtered out of the hollow fiber filters and into the dialysate, so they get removed from the blood. This has been a life-saving, life-extending treatment for many people who have kidney disease. And it's a medical device that works because of a specially designed material that's responsible for its most important function. In this case, it's polysulfone hollow fibers, which keep the blood separate from the dialysate, but allow waste products of metabolism to flow through. So and it was developed by chemical engineers working together with physicians. This is a second device here. This is a stent. It's an all polymer stent. It goes into a blood vessel that's been narrowed because of disease. The stent is expanded when it's in the blood vessel, and then the stent stays behind and holds the blood vessel open. And this stent is mechanically strong enough to hold the vessel open, and it also has a coating on it that contains drugs. And once it's deployed in the vessel, the drugs are slowly released, and they prevent scar formation that can be caused by the presence of this material. And so these are used now in millions of people throughout the world to treat severe heart disease and blood vessel disease. And it's a medical device that's designed with a special material that's responsible for its most important function. And finally, this is a hip replacement. It's an artificial hip. There are two pieces of this. There's a metal piece in this case that goes down into the long bone, the femur, and ends in a ball here, which looks anatomically like your natural hip joint. And that ball fits into a cup that's made of a polymer, and that cup is positioned in the pelvic bones by another metal piece. And this plastic cup is very important for the function of the artificial hip. It provides sort of a lubricated surface so that the ball, when it's moving, as the hip does its normal range of motion, doesn't have metal hitting metal, but it's cushioned by this polymer film. And of course, that polymer film has to be very strong because it's going to hold the entire weight of your body, and it has to last a long time. It's another medical device with a specially designed material that's responsible for its most important function. So these are just three examples that you probably know about, and then you'll know the kind of impact that engineers who have made biomaterials have made in medicine. And these are not small impacts. This is now 10-year-old data, but hemodialysis is used regularly by almost 400,000 individuals in the US. Drug-eluting stents, 4.5 billion in global sales, one of the biggest drug products ever. Total hip replacements, hundreds of thousands just in the US per year. And many of you might be using a medical device now that was produced by materials engineering. This is a contact lens, obviously a soft contact lens being put in contact with one of the most sensitive tissues of your body. Um, and this is an older technology. The idea has been around for a long time. This is a picture from a patent from 1939, where people were using glass as the material to form a lens over the surface of your eye. It was held in place by a binding layer of wax, which doesn't sound so attractive. So contact lenses didn't really take off based on this invention. It took years and material engineers, biomedical engineers, experimenting with all different kinds of materials, including materials that are made from these starting chemicals here, to produce contact lenses that perform the function that we need, that is focusing of light, but also can safely be applied to the eye so that you can wear it for long periods of time without any damage. And this is the balance that we often have to think about. We want a material that functions in a way that we desire, but it also has to be safe and doesn't have to can't cause any unwanted or toxic effects. Because of the work of these people, over 30 million individuals in the US wear contact lenses, and many more throughout the world. Now I'm going to talk about a class of materials that's slightly different than the ones I've been talking about so far. One of the materials is called polylactide coglycolide, has a long history of successful use in medicine. It's an interesting material. It's a polyester. You can make things out of it. You can make sutures out of it, for example, and this is a very commonly used synthetic suture. And it also has the property that it slowly dissolves when it's in water or in an aqueous environment. So if you put this in a test tube full of water, if you put this material in a test tube full of water, or if you implant it into the skin where there's lots of water available, then the material will slowly degrade and disappear. So it's an example of a biodegradable polymer, strong for some period of time and then breaks down slowly. And you can make sutures out of it. You can make devices out of this material. This is a screw that it's used in orthopedics to fix bones in place. And this it'll work just like a metal screw will work, and then it will hold two materials together, and it's mechanically strong. But over time, it will slowly dissolve and disappear as the tissue itself heals. So we've taken that material, and we've engineered it in a slightly different way. We've engineered it so that I'm sorry, I'm trying to. We've engineered it so that it's in the form of small particles. You can see those particles here. They're spherical. They're about the same size, but you can see that there's a distribution of sizes here, and we can modify that by the way we make these tiny particles. You see a scale bar at the bottom here, 2 microns. Each one of these particles, if you measured it, is about 100 nanometers in diameter. 100 nanometers is quite small. It's the size of a virus. Influenza virus, for example, is about 100 nanometers in diameter. And these are small particles that can move through tissues, but totally synthetic. We can totally control their composition. And in particular, we're able to load them with agents so that they have certain properties, which I'm going to show you in the next slide. I'm having a little trouble controlling the screen here, so I'm going to stop for just 1 second. There we go. So here's some of the properties that make these very interesting. One is that the particles themselves are not toxic. So you see in this graph here, we've put these particles in contact with cells in culture and then increase the concentration. And even when you go very high in concentration, the cells don't mind that the particles are there. They're not toxic. You begin to see some toxicity when the concentration is very, very high. If you put drugs inside the particles, so drugs are loaded inside these tiny spheres. This is an anti-cancer drug called camptothecin. The drug, if you make them in the right way, the drug is slowly released from the particles over a fairly long period of time. So this is a period of about 2 weeks here, and the drug is slowly released from the particles. If you put the drug-loaded particles into cell culture, into culture with cancer cells, then as you increase the concentration of the drug in particles, the cells die. So they still continue to act like chemotherapy drugs. Cells will die. But one interesting feature is that the green here shows the drug itself, drug not in nanoparticles. The orange shows the drug in nanoparticles. When the drug is in nanoparticles, it's actually more effective at killing cells at a lower concentration than when it was not in the particles. So somehow drug-loaded particles are more effective at killing cancer cells than the drug itself. And as a result, you can inject these particles into tumors in animals. Here we had a tumor that was growing in the flank of an experimental animal, and we injected particles one time here. And when particles were injected with drug in them, you completely arrest the growth of this tumor. So these drug-loaded particles have very interesting pharmacological properties. These last two observations are related to the fact that the particles, which are green here in this image, are much smaller than a cancer cell. You can see the nucleus of the cancer cell in blue here. You can see the membrane of the cancer cell, which is outlined in red. And after adding particles to cells in culture or to cells within a tumor that's growing in the body, the particles are quickly internalized. They can enter the cell. And now when they're inside the cell, they're slowly releasing the drug, sometimes very near the site of action of drug, which is often in the nucleus for anti-cancer drugs. And it's because of these features, the fact that the particle can carry the drug into cells and then slowly release it near its of action over time. It's those features that are so effective at in this case, killing tumors. Well, one of the other features of having small particles is that they can be administered in a variety of different ways. You can place them on the skin, you can inject them intravenously, you can inject them subcutaneously, you could even infuse them directly in the brain in an approach called convection enhanced delivery, which I'm going to describe shortly. And so I want to just talk about several applications that we've pursued for these kinds of particles based on delivery of drugs for treating cancer, gene editing for treating genetic diseases, and cell targeting to deliver agents to tissues where there's an accessible target. And so I'm going to talk about these three things in order. First, local delivery into a confined compartment or region. And the example there is brain tumors. High-grade gliomas like glioblastoma, a very aggressive disease, a universally fatal disease, even with very aggressive treatments. It's not that rare. Tens of thousands of patients are diagnosed just in the US, and survival is poor. So the standard of therapy is to do surgery, if surgery can be done, to give radiation therapy after surgery, and chemotherapy together with the radiation therapy. And even with that multimodal treatment, the average survival is about 15 months. And you can see that on the curves here from the New England Journal of Medicine. This was an article from 2005 when they just introduced radiation therapy plus the drug temozolomide. You saw a significant increase in survival of a couple of months, but nothing has changed it since this time. We began working on this many years ago, thinking that materials might be useful for delivering drugs to the brain, and the kind of materials we chose were these degradable polymers. We didn't make them into nanoparticles at first. We made them instead into wafers that are about the size of a dime, and you can see it in this photograph here. But they were designed in the same way that I showed you before, so that the drug that's inside this material is slowly released over a period of several weeks. And we designed these to be used as a tool by neurosurgeons. So when they were treating a brain tumor, they could identify where the tumor was. They would do surgery to remove the bulk of the tumor, and then they would place these wafers into the surgical resection cavity so that after surgery and the patient left the operating room, these wafers would be slowly releasing the drug compound, and it would penetrate into the tissue around the tumor. And the rationale for this is that the surgeons know that they can remove much of the tumor surgically, But what you can't see here in this radiographic image is that there are individual tumor cells that have migrated out into the tissue that's around the bulk of the tumor. And they can't remove all those cells because they don't know where they are. And there's lots of normal tissue there as well. And so it's these tumors, it's these cells that have migrated away from the bulk of the tumor that are causing tumors to reappear and the reason that the disease isn't treated effectively by surgery. So the idea of these polymers is that they would release drugs into this region around the tumor and hopefully treat that residual tumor cell mass, those residual tumor cells after the surgery. This concept was tested by Henry Brem, led a clinical trial of this in the 1990s and found that in a placebo-controlled trial that patients that got the polymer with the drug BCNU lived on average longer. And there was a statistically significant increase in long-term survival here. And this is what led the FDA to approve this approach in 1996. Still, it's not a cure. It extends life, extends dramatically life for a subset of the patients, but not for all. And so we've tried to explore how to do this even better. And one of the limitations of this approach that we discovered while we were developing it is shown in this figure down here. If you place one of these wafers in the brain of an animal and you do it with radio-labeled drugs so that you could track where the drug is over time, you find that, that large quantities of drug are released, lots of drug is released into the brain, but none of it penetrates very far into the brain. It has to diffuse through tissue, and as it diffuses, it disappears. And so the end result is that you're only treating a region of about 1 to 2 millimeters from the polymer when you do this. Now, drugs are released over a long period of time, drug concentrations are very high, but it doesn't penetrate very far, and so the volume you can treat is limited. If a tumor cell has migrated away from the tumor and is out here, maybe only a few more millimeters away, it would be resistant to treatment. We've pursued with a neurosurgeon, Joe, before, the idea of polymer implant. It was shown to be effective in a certain cases. At the same time that we were developing this enhanced delivery. And in this approach, people put a catheter here is that if you do this properly, if you infuse a drug in solution, it will slowly spread through the brain. And as you add more drug, it spreads further and further and further. And so you can control the volume that you treat by how much drug you infuse. The problem is you can't do this infusion for very long. You have to be in the ICU. It's a dramatic procedure, so you can only do it for a day or so. And if you remove the catheter, then the drug all disappears. There's no more drug. We thought you might be able to combine the benefits of these two approaches by using, instead of a drug wafer, using the nanoparticles that we had developed with anti-cancer drugs in them, infuse them through the catheter in the same way that's done with convection enhanced delivery of drugs in solution. But now you'd be spreading nanoparticles over the brain. You remove the catheter, the nanoparticles are distributed, they stay where they are, and they release drugs slowly. They would function like this implant, but they'd be spread over whatever volume the surgeon chose. And so you could treat a larger volume of the brain. And our first experiment to test this idea was done in animals that had brain tumors. And here this is a tumor that once initiated in the animals will die within about 2 weeks. If they're treated with just drugs infused into the brain, it doesn't do very much. One animal lived a long time, but most of the animals still died after 14 days. But if you deliver the right dose of nanoparticles this way, you can increase the median survival, and you got about a third of the animals who lived a very long time. If you go look in the brains of these animals, there's no evidence of tumor. So we were quite pleased with how this worked. But when we looked in the brains of the animals, we found that the nanoparticles themselves didn't go very far. This is where the catheter was in the brain. The magenta region here is the particles that we delivered. They all accumulated around the tip of the catheter and none penetrated into the brain the way that we predicted. Well, the problem is these were particles that were about 150, 160 nanometers in diameter. We thought they'd be small enough to penetrate through the brain, but we were wrong. They didn't penetrate very well. They just stayed around the end of the catheter. So if that's the problem, if the problem is the particles are just slightly too big, then the solution is simple. You just make them smaller. And we learned how to make them smaller. It took several years. Because remember, we don't just want to make small particles. We want to make small particles that are highly loaded with drugs. And that just turned out to be technically difficult. You also, as you make particles smaller, you increase their potential for aggregation. And we had to figure out ways to keep these very small particles from aggregating. But once we'd solved those problems, once we'd made them smaller, once we'd improved them to make them resist aggregation, we found that we could infuse nanoparticles in the brain and they would distribute throughout the hemisphere of the brain of this animal. We call those brain penetrating particles, and they allowed us to think about this treatment in a completely different way, because once you have particles that you can administer to the brain and you can get them over large volumes, then the challenge of treating tumors is simply finding the right drug to put in the particles that's most effective at killing them. And I had a postdoc named Zhongbing Zhao, who's now on the faculty at Yale, and he discovered, using a variety of approaches, an FDA-approved drug called dithiazidine iodide that was remarkably effective at low concentration at killing the most aggressive population of cells within a tumor. Those are called glioma stem cells. He loaded that drug into these nanoparticles, and he did it in a way such that the drug was slowly released over a very long period of time. Then tested these in one of the most authentic models of human glioblastoma in experimental animals. This is created by injecting human glioma stem cells into immunocompromised the brains of immunocompromised animals. If you don't give any treatment, then the animals die in about 180 days. If you give a standard treatment, that is infusing free drug, they live a little bit longer. Drug and particles that are too big to penetrate, about the same, but drug that's in these brain penetrating particles that can move throughout the whole hemisphere of the brain, you see a remarkable degree of effectiveness where almost 75% of the animals live a long time, no evidence of recurrence of tumor. So we're working with our colleagues at the Yale Medical School to try to translate a new approach for treating tumors into clinical practice. The second thing I want to talk about is systemic delivery of agents that are targeted, so that delivery to non-target cells is irrelevant. And in this case, I'm going to think about intravenous injection of nanoparticles that contain gene editing agents. And the gene editing agents that we've worked with are called triplex forming oligonucleotides. These were, this approach was pioneered by Peter Glaser, my colleague, who's a physician and a scientist at Yale. I mentioned before, he's chair of the radiation oncology department, so and an expert in DNA repair. How does DNA repair itself when it gets damaged? And that damage to DNA occurs in cells throughout our lives. Most cells are very good at repairing small defects in DNA. And he's exploited those repair mechanisms by designing molecules called peptide nucleic acids that can bind to DNA in a site-specific way. And when they do that, they form a triple helix. And that formation of a triple helix is part of the design of the PNA. You design a PNA such that it binds at a specific site in a gene. Say this is a gene in the sickle cell gene, and you want to repair the sickle cell gene because there's a mutation. And so you design the PNA to bind somewhere within that gene. Form a triple helix on binding, and it evokes natural repair mechanisms. The cell tries to repair the DNA by activating DNA repair pathways. We also, Professor Glaser learned that if he delivered the DNA together with the PNA, together with a donor DNA, that is almost identical to another site in the gene, but contains maybe a few differences. So almost exactly like this template in the genomic DNA, but say has a few 1 or 2 or 3 bases modified, that sometimes the repair mechanisms inside the cell will use this synthetic template rather than the natural template, and will create a permanent change in the gene that you could detect using biochemical methods like allele-specific PCR. The result is you deliver these two agents into the cell, and sometimes you invoke a recombination and a gene edit that's permanently inheritable at this site. The challenge for this approach is how to get these two molecules into the cells that you want to do the gene editing in, how to get them safely inside the cell without destroying the cell. And these molecules obviously have to get into the nucleus in order for the process to work. So we took these PNA and DNA molecules, and Dr. Glaser and I started collaborating together about 10 years ago. I should mention too that over the 10 years we've been working on this, with other chemists like Danith Lee at Carnegie Mellon, have made improved PNA molecules that are even more efficient at binding and forming triple helixes. Our role in this research has been to develop delivery systems that could get these molecules inside cells. We started off using simple systems like the ones I've showed you before, where we make nanoparticles that incorporate the PNA and DNA molecules into the structure of the nanoparticle. We can then deliver these nanoparticles to experimental animals. Here's a number of publications that we've produced over the last years showing how this approach works. Well, I'm going to speed ahead over many years of work and give you one of the end results here. This is a mouse that has beta thalassemia. That beta thalassemia in the mouse, it has the human beta globin gene, which has a common mutation that causes beta thalassemia. It's a mutation in the beta globin gene that leads to anemia in the animal and affects throughout the animal's body because of that anemia. So this mouse was genetically engineered so that it contains the human gene with the defect. Peter Glaser and his laboratory designed PNA and DNA molecules that could repair that gene. We loaded it into nanoparticles. We gave these mice 4 injections of nanoparticles over a period of about a week and a half, and then we monitored them. The animals that don't get the injection or get injection of blank nanoparticles remain anemic throughout their lives. And you can see that because their hemoglobin levels remain low. It should be up in this region here, but they're anemic. They don't have in their blood, so that a PNA DNA cells, hematopoietic stem cells that are characterized by these markers, we edit the highest amount. So we think this editing in the bone marrow is what's responsible for this correction of anemia. Reticulocytes and blood cells go down dramatically with treatment. The size of the spleen, which ordinarily these animals get very large spleens because of their anemia. The size of their spleen goes back towards normal, and the histology of the spleen starts to look more normal as well after treatment. So we call this something like a cure beta thalassemia by just injecting 4 injections of nanoparticles. How early in life could you apply kinds of gene repair approaches? Because it's known in diseases like beta thalassemia and sickle cell anemia and cystic fibrosis, because of the genetic defect, there's changes in physiology throughout the body, and people get sicker as they live with this defect. So there's a big advantage to being able to treat early in life. In fact, maybe you could treat before birth, because many genetic diseases can be diagnosed early in pregnancy. And there's approaches like cell-free fetal DNA tests on mother's blood that can identify many genetic diseases. Currently, fetal surgeons can intervene on a fetus as early as 12 weeks of gestation under ultrasound guidance. And so they're doing things like fetal blood transfusions now to treat certain kinds of conditions. So we think genetic correction in utero before birth, early in pregnancy, could potentially treat and cure numerous diseases and allow normal fetal development. We worked with a pediatric surgeon who is skilled at fetal surgery, named Dr. David Stittleman, and he came to Yale a few years ago and has the remarkable ability to be able to microneedles into the blood vessels of animals while they're in utero. So this micro pipette that's filled with nanoparticles, and you can see that micropipette is going into the vitelline vein of a fetus that's within the uterine horns of its mother, a mouse. And he does the injection, and you'll see these nanoparticles entering the vein, and then they're distributing in the circulatory system of the fetus. We did the same, if you deliver these fluorescent nanoparticles to a mother at different times, you can see the particles which are lit up here in this image. This is an IVIS image, an in vivo imaging system that allows you to see fluorescence inside the animal. These are all the pups that were treated with nanoparticles in utero. After those animals are born, there's fluorescence left in the animal. You're giving the treatment to fetus. And you could give it at different times during gestation, either injecting intravenously, as I showed up here, or just injecting into the interamniotic space in an approach that's widely used for amniocentesis. If you look inside the fetus itself, you can see nanoparticles all throughout the tissues of the body, in the brain, in the heart, in the lung, in the gut, but particularly in the liver. And this is very interesting to us because in fetal animals, hematopoiesis, the creation of new blood cells, doesn't occur in the bone marrow, it occurs in the liver. The hematopoietic stem cells are within the liver during this stage of gestation, and then they migrate out to the bone marrow at a later stage. And so we thought if we can intravenously in animals like this, a lot of the particles would go to the liver, and potentially they would treat these hematopoietic stem cells. In fact, that turned out to be true. This is from the PhD thesis of Adele Ricciardi. She injected either blank nanoparticles or particles at increasing concentration. You see hemoglobin levels here. Animals that don't have the mutation that causes beta thalassemia have normal levels. If you treat with a high enough dose of nanoparticles with one injection during intrauterine life, you correct the gene, you reverse the anemia, and those hemoglobin levels remain high throughout their lives. The size of the spleen goes down, and the morphology of the spleen starts to return to normal, and the reticulocyte count goes down as well. So this is with one single injection in utero, an approach for correcting genetic disease that causes disability and mortality throughout the world. I'm going to give you one last example. I'm going to call it targeted delivery, but it's targeted in a very particular kind of way. One of the things that we learned was that not only size and shape of these nanoparticles as to their function, but the properties of the surface can also have a big effect on how the particles work. And so this is a very interesting particle that we developed that has a core that's a biodegradable polymer. It's polylactic acid. And the surface of it is a hyperbranched polyglycerol, or HPG. And hyperbranched polyglycerol is a very water-soluble molecule with lots of hydroxyl groups on it. And so it gives the particle a stealthy quality. It reduces their sticking to each other. And if you inject these in the bloodstream, they circulate for a very long period of time because they don't stick to anything. They're very non-adhesive. But interestingly, because of the chemistry of this hyperbranched polyglycerol, where the vicinal diols here are converted into aldehydes. And this is a simple treatment with sodium periodate accomplishes this chemical change. And now they have aldehydes all over the surface. And aldehydes are very sticky, particularly sticky to proteins. And we can show that here. If we take these stealthy particles, non-adhesive ones, and we suspend them over a surface where there's protein surface, they don't stick. You can off. These are fluoride particles. You can see them here, and you can wash them all off, so no fluorescence is left on the surface. If we convert them to the sticky form and put them on the same surface, protein-rich surface, they stick. You can't wash any of them off. Same thing with cells in culture. If you put the stealthy particles on cells in culture, they don't stick. You can wash them all off. But if you put the sticky particles on cells in culture, they stick. You can't wash any of them off. Well, what would you do if you had sticky particles like this that you can now target to a location and they will stay there for a long period of time? We decided we could use these to make an improved sunscreen or sunblock that's based bioadhesive nanoparticles. And we do that by loading into the polymer core, the hydrophobic core of this polymer. We load with UV filters, molecules that absorb ultraviolet light. And these are the same kinds of molecules you find in bottles of sunscreen that you would purchase at the drugstore. We take those molecules and we put them into adhesive particles. And when you put them into adhesive particles and apply them to the skin, they stick to the skin. They spread all over the skin and they stick and they stay there. And if you try to wash them off, you can't wash them off. And I show that here. If you put them on the back of a mouse in this case, the particles are fluorescent. You then let the mouse swim or you wash it with water. They don't come off. If you take a towel, a wet towel, and rub it over the back of the animal, you can remove almost all of the particles. If you don't do that, but you just let them run around in their cage, the particles will stay on their surface for many days. And that's shown in this graph down here. We can apply them to skin and they have ultraviolet filters in them. They absorb light and they prevent sunburn. And they also prevent the kind of DNA damage that happens within the skin when skin is exposed to ultraviolet light. So if you have no protection, no sunscreen, create these DNA breaks in the DNA that can be identified by staining with cyclopyrimidine dimers, and you get effects of sunburn here that you see histologically as well. When we put our particles on the skin, it absorbs that ultraviolet light and prevents that kind of UV damage. So we're working hard to develop this technology into an approach that could be used to provide long-lasting, more durable, and we think safer protection of the skin from ultraviolet light. In summing up then, this is a field of nanoparticles viewed by scanning electron microscopy. I've tried to show you that nanoparticles can provide sustained delivery of agents when administered locally. They can provide intracellular delivery of complex agents in tissues throughout the body, and that new formulations of nanoparticles allow for targeted delivery in situations where there's an accessible target. We think these could be useful for treating brain tumors, as I've shown you earlier in the talk, that you could use these for gene editing in situations like cystic fibrosis, and we've published a few papers now showing that this approach can be used in cystic fibrosis. And they could prevent what I didn't talk about. You can use them to deliver antiviral agents to prevent virus transmission like HSV, and that they can potentially be used for effective, longer lasting sunblocks. And this could affect millions of people throughout the world. Well, when I give talks like this, I frequently get the question, do Yale undergraduates work in your lab? And the answer is yes. And so this is a photograph of my lab group from the summer and the past summer. And I'll just point out a couple of people here. Elias Quijano, who I mentioned before, started working when he was between his freshman and sophomore year. He graduated in Yale College in 2012. He stayed on as my lab manager for 3 years, and he was fabulous in that role. And now he's in the MD PhD program at Yale. And in the PhD phase of his program, he's been working on these gene editing agents for treating sickle cell anemia. So he started off right after his freshman year, and he's been a very productive research scientist since that time. Hannah Mandel graduated in 2017. She's been working in my lab for a couple of years and is on her way to medical school at UCLA starting in the fall. Kei Nakazawa also graduated that year and worked in my lab and now is studying in graduate school in the Bay Area. Jenna Dorito started as a freshman working in my lab and just completed her PhD at Cambridge University in the UK. Rachel Putnam is graduated in 19, spent an extra year in the lab and will be starting medical school at Duke in the fall. Allison Ho is graduating this year. Krista Chen is graduating this year. So the answer, do Yale undergraduates work in your lab, is yes, they do. Many of them do, and they contribute in fabulous ways. I thank you for your attention, and I'm available to answer questions. Let me see how I get the chat open here. Should I stop sharing? Okay, there's a question from James. Won't the material become decomposed by the blood's acidity over time? Yes. Well, the blood is normally neutral. It's not acidic. Its pH is about 7.4. They do degrade over time, and it depends on the chemistry of the material itself, what the exact composition of the polymer is. But the kinds of materials I've shown you here, they will degrade over a period of 6 weeks to 6 months, something like that, depending on how they're designed. Rohan asks, is there the potential for these particles to deliver drugs based upon physiologic changes in the body like pH or glucose value? That's a great question. So Rohan is asking, can you make the particles responsive to physiological change such that they would get activated, let's say, when something happens, like say there's a pH change. And we have done work on that pH, and one of the applications of pH is that many tumors, solid tumors, have a lower pH than normal, so they're at maybe pH 6 instead of pH 7.4. And we've designed materials that can accumulate only in areas where there's low acidity. If you want to write to me, I'm happy to send you a paper that describes that. Having ones that respond to glucose would be very, very valuable. For example, if you had a polymer that released insulin, a particle that released insulin only when glucose levels were high, then that could be a very self-regulating system for treating diabetes. And people have been working on that for a long time. I've not on it personally, but it's turned out to be a very challenging problem to solve. How far out are we from the use of nanoparticles in human trials? That's a great question too, and it depends on the application. So with the sunscreen, which I talked about, my colleague Mike Girardi, who's a dermatologist who I did all this work in collaboration with, we actually ran a pilot clinical trial to test these and compare our particles to a conventional sunscreen. We just did that in a handful of patients, and we found that they were very effective as the normal sunscreen, and importantly, no side effects were seen when we applied these particles to the skin, and so that's very important. So there's a first application, first clinical trial in humans, and we're hoping we can do more in that way. Now that's because you're applying the particles topically. That's a little bit easier to do than injecting them systemically. From a safe perspective, it was easier for us that clinical trial. To do gene editing is going to take more work, and I think that those kinds of applications are still several years away from clinical trials. We're working very actively to try to move in that direction, both for treating cancer with particles, as I showed you at the beginning, and with gene editing. Have you seen any risks or side effects associated with the use of nanoparticles? So there can be risks. It typically it's not been associated with the nanoparticles. The particles themselves, when they're not loaded with drugs, seem to be non-toxic in most situations. Now, we have been synthesizing new versions of polymers that can be used in making nanoparticles, and some of those do show signs of toxicity. So we know we have to be careful about making sure that we design the material so that it is totally inert. Most of the side effects come from the agent that you put inside it, the drug, a chemotherapy drug, for example. So one has to study that. And we have to be very thoughtful about how to administer them and administering them in ways where we know that we're at a dose that's safe. Vanessa asked, do you welcome undergrads with experience in your lab? Most of the ones I mentioned who started early in their careers had little or no laboratory experience. And it's very important for me to pair up undergraduates with mentors who've been in the laboratory for a while, usually graduate students or postdocs, and pair them up with people that know how to do the techniques that they're interested in learning how to do, but also pair them up with people that I know are good teachers and are going to be able to provide a good experience for someone who's new to the laboratory. So I try to do that in a very thoughtful way, and because of that, that sort of limits the number of people we can do, because we have to set it up like a learning experience where someone is dedicated to making sure that training you need to work safely in the laboratory, and also develop skills so you can collect good data. James asked, how easy is it for those AP classes in subjects such as biology, chemistry, and or physics to take biomed classes early on? It is possible to do that. I think I've had first year students who've taken, for example, the physiology class that I mentioned. I certainly have had a lot of first year students who've taken the introduction to biomedical engineering class that I've offered. The core of the biomedical engineering curriculum really starts during the junior year. And you really do need to have physics and chemistry and some biology before you do that. So it's harder to get into those classes early on, but it's certainly possible to take, to engage with some of the material. Maya asked, does the engineering of nanoparticles to treat tumors change depending on the location of the tumor? That's a great question too, and it does. I've showed you one example where we're delivering them directly into the brain, and there we found that we needed to make the particles much smaller than we would if we were delivering them somewhere else. And so that's one change that we had to make in order to introduce them to that special environment. If you're going to introduce them intravenously, then typically you make changes in the particles such that they will accumulate in the tumor rather than accumulate in other places. And there's a variety of ways that you can do that by manipulating either the size of the particle or its surface chemistry. Vanessa asks, are the drugs attached or inside the particles? It's another great question, and you can do it either way. The ones that I've shown you here, the drugs are all inside the nanoparticles. They're within this core. There might be some that are near the surface, but the bulk of them are in the core, and the way we make them, that's what we're trying to achieve. You can also chemically attach drugs to the surface. Typically, you would get a much lower concentration of the drug if you just attach it to the surface rather than putting it throughout the material. So we've tended to use the surface to try to direct the particles to either adhere to particular cells or to target cells. Ryan asks, can nanoparticles be designed to encapsulate more than one drug? They can, and we've published some papers about that as well. Combinations of chemotherapy agents are particularly interesting. You've seen in the example with gene editing, there really were two different molecules that were put into the particle at the same time. And we tried it several different ways. We tried making, remember, it's the peptide nucleic acid PNA and the template DNA. And in our early experiments, we tried it putting the PNA and DNA in one particle. We tried putting the PNA in one and the DNA in another. And it turned out that when they were both in there together, that worked better. And so we've stuck with that approach, but it is possible to put combinations of drugs in. Maya asks, also, for how long does it usually take to get FDA approval for a particular drug? Maya, that's a great question. I wish I had an answer. And it depends on the situation. We think with the sunscreen, we can get approval relatively quickly because the FDA has already approved the agents that you would use inside. There's a list of sunscreen agents that are already approved by the FDA, and all we're doing is delivering in a different fashion. So we just have to prove that that approach is safe, and then we can potentially get FDA approval quickly within, we think, a year. For other things, it's going to take much longer, particularly in cases where the drug itself has not been approved for some other use in some other delivery system. So in the gene editing approach, for example, those agents haven't been approved for anything yet, so that's going to take a longer time for the FDA to feel comfortable that everything is safe and effective. So that would be years. Kahil asks, has your lab done any work in associating imaging proteins or compounds to the nanoparticles? So that's another great question, and we've used a variety of imaging approaches. In fact, for the agents that we infuse into the brain, we can make them visible to magnetic resonance imaging, and we've used that so we can track where the particles go while they're being infused. We've also worked with our colleagues at the Yale PET Center to put positron emitting isotopes on the surface of the nanoparticles so that we can image them by PET imaging, a nuclear medicine approach. And that turns out to be very sensitive and quantitative, and you can follow the particles over time very easily. We're doing that now, and we're to track particle fate in the brain after we introduce them. James asks, for trials such as those used in the cranial cavity, do the nanoparticle capsules ever degrade or remove from the cavity after the drug has been administered? They remain indefinitely. So they degrade. As I mentioned before, you can control that to some extent by controlling the chemistry of the material. It's a little bit hard to study because we could study that very carefully when we were doing the dime size implants, because you can put them in the brain and then you can go back later and you can see if it's still there. You can take some of the material out and you can measure its molecular weight. With the nanoparticles, they get dispersed into tissues and inside cells, and so you can't easily recover them and ask how much is degraded. So we do complementary studies where we just put them in test tubes full of water, and then we study it there. We control the pH, we control the temperature. Sometimes we do it in artificial cerebrospinal fluids, so we're mimicking the brain chemistry to some extent, and then we can watch how they degrade there. And in that case, it takes several months for them to completely degrade. I think I'm getting the message that was a question to end on, and I thank you for your attention. I hope that I will see you in New Haven soon. I hope I've convinced you that particularly for someone like me who's interested in engineering and medicine, Yale is a fabulous place to be, fabulous place to learn, fabulous place to teach and to study. So I hope I'll see you here soon. Bye.